Hello, this is Melissa Davis at the GRU-UGA Medical Partnership, and this is the podcast for Modes of Inheritance 1. In this podcast, we'll be discussing Mendelian genetics, gene expression, and gene mutation. Listed here are the learning objectives of both this podcast as well as the large group session that you'll be a part of in a later date. Essentially, what we're going to do is discuss some basic laws of inheritance that were established by Gregor Mendel, the law of segregation and independent assortment. We'll also distinguish among different patterns of inheritance. We'll discuss how phenotypic behaviors can impact the inheritance patterns within families. We'll also deduce how different DNA mutations, different types of mutations, can impact gene expression or gene functions. And we'll compare and contrast expressivity as well as penetrance within a pattern of inheritance to discuss how this can impact interpretation of a pedigree. Gregor Mendel has been noted as the father of genetics because in his work he was basically observing the different phenotypes of specific traits in pea plants, from seed coat to seed color, pod shape, seed shape, stem height, as well as flower position. In his observations, he basically tracked how different phenotypes segregated with other phenotypes depending on what the parental phenotypes were. While he didn't realize it, basically what Mendel was observing is something called chromosomal segregation. In his law of segregation, he noted that different phenotypes segregated in a certain way. Essentially what he was observing is the fact that each individual has 2 copies of a chromosome. During replication of interphase, we have duplicate chromatids of each chromosome, and during meiosis, these chromatids are all separated into individual gametes. So depending on the original genotype of parent, that will dictate what each one of the gametes will hold. But only 1 copy of the chromosome will be in each gamete. In this way, we segregate the alleles into the gametes. Another one of Mendel's important laws was the law of random or independent assortment. Basically, this law just takes account of the fact that certain traits are inherited without regard to others. This is because the genes that are responsible for these traits are not linked. In other words, they don't reside very close together. Independent assortment also occurs during meiosis. So for this particular schematic, we're looking at 2 different traits, or 2 different alleles, the A allele and the B allele, or 2 different genes, the A gene and the B gene. And there are 2 different alleles of each gene, the big A allele and the little a allele, the big B allele and the little b allele. Throughout metaphase and telophase of meiosis 1, we have separation of the chromosome homologs. As you can see, depending on how the chromosomes are separated, you might have separation of all of the wild-type alleles or separation of all of the mutant alleles or a combination of both. Fast forwarding to the end, after telophase 2 of meiosis 2, that means that we can have different combinations of these traits in the gametes. And this assortment of whether the dominant or the mutated allele are within the same gamete happens randomly. Now let's discuss patterns of inheritance. First, let's discuss autosomal inheritance. Human autosomal traits simply mean that the genes that are responsible for the trait reside on non-sex chromosomes, which are chromosomes 1 through 22. They may be inherited in either a dominant or a recessive fashion. Autosomal dominant traits basically are transmitted by males and females, and we do see male to male transition occur. And we see in the case of dominant traits, we usually see successive generations that are affected. And transmission usually stops after a generation in which no one is affected. So the mutated allele has not been passed on and therefore will not be seen in the family any longer. In recessive traits, again, both males and females can be affected. And both affected males or females can transmit the gene and will transmit the gene unless it causes death before reproductive age. In recessive traits, we usually see that the trait skips a generation. However, there are usually carriers in that generation that we'll see in their offspring, that where we will see the phenotype or the trait reappear in their offspring. Parents of affected individuals are by definition heterozygous or carry the trait. So if we look at these two examples, an unaffected parent, who in this case has two small A alleles, versus an affected heterozygous parent who has one big A and one small a, the offspring of which will be half affected and half unaffected. In this case, we usually say that the heterozygotic situation will display the phenotype, and that's another way to define a dominant allele. In a recessive trait, you actually have to have two copies of the mutated allele in order to see the trait. In the case of recessive alleles, in this case we have two unaffected parents who are carrying a curly and a straight allele. In this situation, the affected child has to have both copies of the straight allele in order for us to actually see the phenotype. In this way, we know that this is a recessive trait. In families where there's a high level of consanguinity or relatedness between couples, we usually see an increase of recessive traits. X-linked dominant versus X-linked recessive traits are unique in that, first of all, they reside on the X chromosome. And so as we know, males do not pass on X chromosomes to their son. So in this case, we usually don't see a transmission of male to male traits, whether it be a dominant or recessive trait. In X-linked dominant traits, females will transmit the trait with higher frequency, and successive generations are affected. In X-linked recessive traits, affected males will receive the trait from their mothers, who are carriers of the trait. In this example, we see a pedigree of a family with faulty enamel. We already know that this particular trait is caused by a mutation that is on the X chromosome. Through the information from the family history, which we can see already is somewhat incomplete, we are tracking the fact that the female in the first generation passed it on to a daughter, who passed it on to her son, at least one of her sons. And here is where we are able to now determine the sex-linked nature of the disease. This son has passed on the trait to all of his daughters and none of his sons. Likewise, his daughters have passed it on to a son and a daughter, and in this case a daughter. And so we are able to deduce that this is an X-linked trait. Here I just simply list the inheritance of some common traits that you're aware of. So widow's peak, freckles, cleft chin, and dimples are all dominant traits. Straight hairline, clear skin, rounded cheeks, and rounded chin are actually recessive traits. I usually joke that my daughter has gotten the worst of everything, as I have freckles, her father has a widow's peak, and her father has a cleft chin. And so she has all of these traits. Here I simply list 5 general steps that will help you in solving genetic problems. The first thing you should do is try to list all the genotypes and phenotypes for the trait. Then determine the genotypes of the parents involved. This will help you to derive the possible gametes of the offspring. Then when you unite all possible gametes and all combinations, that will reveal all possible phenotypes. And then just repeat that for each successive generation. You'll find that it will be very beneficial to use a pedigree when the problem requires that you identify the pattern of inheritance, especially for a trait that you do not have any prior knowledge of inheritance. Now we'll switch gears and we'll start talking more about the molecular nature of genes, the structure, expression, and mutation. So now we meet the chromosome. The P arm versus the Q arm. The Q designates the long arm. The P, which stands for petite in French, represents the short arm of a chromosome. We designate each side of a chromosome so that we can identify or map the location. The numbering that occurs basically represents the banding patterns that we see in chromosomes when they're stained with Giesma dye. We'll discuss more about chromosomal structure and chromosomal aberrations in Module 6. Depending on where a mutation occurred, it can severely disrupt a gene or have very little impact. All of these locations are typically found in clinic. You can have a mutation in the promoter, which basically shuts on or off the actual expression of a gene. You could have a mutation in a key site for a key site of translational start. Depending on where a mutation occurs in the gene, this can severely impact the expression of a gene. To keep this simple, basically you can have genes that impact the expression levels. You can have mutations that impact the gene's ability to be processed. And then lastly, you can have genes that actually impact the gene product. New mutations, especially in germline cells, are basically the birth of new alleles in our gene pool. Those that occur in somatic tissues are usually spontaneous and are held within the individual where they occur. Those that occur in the gene line can be passed on, and these are usually inherited. Mutagenic factors that alter DNA, of course, include radiation and chemicals. But more importantly, the types of DNA mutations that occur, such as insertions, deletions, and substitutions, can alter the function of a gene. Mutations are usually classified by their effect on the gene product. We can dichotomize this classification by calling them generally loss of function or gain of function. This table is here for your reference, and basically all I want you to take away from it is that in loss of function genes, they occur pretty frequently, where you have a mutation that removes completely the function of a gene, or something we call hypermorphic or leaky, meaning that it impacts the function but it doesn't remove it completely. Usually loss of function genes are recessive. This is because you have two copies of the gene. So if you remove the function of one, you still have the copy of the other. In cases where recessive mutations display as dominant, this usually indicates what we call haploinsufficiency, meaning there could be a deletion of that particular gene on the other chromosome. In the case of gain of function, these are usually very rare. And what happens is that either they are hypermorphic, meaning that there's too much expression of the gene, or they are neomorphic, where there is expression of the gene in a region where it normally does not express. Mutations in the coding sequence of a gene can alter the gene product. such as nonsense mutation, which changes a codon that usually brings an amino acid into the protein into a stop codon. This usually truncates the protein. Frameshift mutations basically change the sequence of amino acids and the actual content of amino acids. More frequently, we see silent or synonymous mutations, where there's a change in the DNA sequence, however, the amino acid sequence has not changed. This schematic basically gives you an example of what a missense mutation is, a nonsense mutation, and a frameshift mutation. In the case of sickle cell, this is caused by a point mutation. That is a a non-synonymous substitution. In this way, the single amino acid changes a glutamine to a valine. This alters the chemistry of hemoglobin such that instead of being in rounded globule and rounded molecules, it attaches to itself into fibers. These fibers elongate within the cell and actually stretch the cell out of shape, giving it the sickling shape, hence the name of the disease. Some clinical consequences, of course, to sickle cell are pleiotropic. We'll discuss more about pleiotropism in class, but basically what I want you to take away from this diagram is that depending on where the clot may appear, as well as the state of the individual at the time, so whether oxygenated or deoxygenated, the impact can either be severe, from acute pain, chronic with neuropathy, chronic with inflammation, or chronic with hypertension. Thank you for your attention, and I'll see you in class shortly.